Welcome to Cancer Newsline, a podcast series from the University of Texas MD Anderson Cancer Center. Cancer Newsline helps you stay current with the news on cancer research, diagnosis, treatment, and prevention, providing the latest information on reducing your family's cancer risk. I'm your host, Lisa Garvin, and today our guest is MD Anderson President Dr. Ronald DePino, and we're going to talk about a momentous occasion in MD Anderson's history, the Moonshots program. Welcome, Dr. DePino.
Lisa, it's great to be here. Thank you.
And I guess the first question is, what is a moonshot?
In a nutshell, it's a comprehensive, action-oriented effort across the entire cancer care continuum designed to dramatically reduce the mortality in that cancer in the near future and set the stage for ultimate control and cure of the disease.
I think some people would argue that that's the end goal of cancer, period. So what defines a moonshot as opposed to any other efforts at eradicating cancer?
Well, the guiding principles are as follows. It sounds more or less like another strategic plan, but this is very different. And perhaps it's captured in the idea that when Kennedy said, we're going to go to the moon, he didn't say we're going to study as to how to go to the moon. It was a real goal. So a very important aspect of this plan is to try to think in terms of achieving deliverables that would really impact on patients, on their survival, on their quality of life. And if you wanted to go and cure a cancer, you soon realize that what you need is to fire across the entire spectrum of clinical care activities, from prevention to early detection, prognostication, which means improved management, treatment, and ultimately survivorship. So one aspect of this is our ability to fire across the entire beachfront of activities that really prevent the occurrence of the disease in the first place, or if it happens, catch it early, or if it does occur, to be able to wrestle that disease to its knees. The other very important aspect of this is that we're going beyond discovery. That is to say that we have the development of enabling platforms that are helping our brilliant clinical and basic researchers reach diagnostic and development endpoints. So it's about going beyond discovery and doing. It's not enough to know. We must actually apply that knowledge and reach a tangible endpoint that impacts on patient survival and quality of life.
And I understand the underpinning for this is what's being called the discovery platform. And I guess it's a collaborative effort to kind of bring a lot of statistics, a lot of data, a lot of researchers and clinicians together. Is that correct?
Yeah, so one aspect of this, again, a defining principle is the integrated nature of this particular platform. And I'll give you an example of this, of how different disciplines have come together to solve a particular problem. And the other aspect is also instilling this backbone of directed activity that allows us to go from A to Z and really achieve these clinical endpoints in these professional platforms that we'll be implementing. So with respect to integration, one of the very exciting things that I've learned is in the area of lung cancer detection. And so we know that there are millions and millions of heavy smokers that are at risk for the development of cancer. And we now know that spiral CT will allow us to detect those cancers earlier. And as a result, in the first studies, we now can achieve a 20% reduction in mortality. However, we have to screen many folks, and there is a 96% false positive rate. And so in thinking about the goal of how to do this better, we had folks from many different disciplines come together and actually bring their knowledge and capabilities to bear on the problem. We have here at MD Anderson developed a fantastic risk model. That is, we know amongst those heavy smokers which 10% or so are knocking at cancer's door. And we can enlist those individuals into aggressive screening and avoid having to screen unnecessarily many others. In addition, with that 96% false positive rate, it means that we're going to be chasing a lot of ghosts on CT. That's also going to drive a lot of issues with respect to management, costs, and so on. And one of our great new recruits, Sam Hanash, who's one of the world's experts in early detection and serum biomarkers, has a very interesting marker panel that may allow us to detect cancer much earlier than onset of clinical symptoms, which means now if we can triangulate with the imaging plus the blood test, we may be able to improve the accuracy of detection and also reduce the false positive rate for those that are harboring the cancer. So here again, we have three different groups that came together in prevention with risk modeling, in our imaging program, which is second to none under Marshall Hicks, and then we've got folks in serum proteomics like Sam and others that are coming together to really solve a problem in a multidisciplinary, multi-technology way. Very exciting.
And I think people would be surprised that actually people tend to work in silos in academia. You've got your researchers in the lab, you've got your clinicians. So it's kind of speeding up the translational nature from the lab to the clinic.
That's exactly right. I think for centuries in academia, we've celebrated the individual. You think about the greats, Madame Curie or Louis Pasteur, etc. But science has just gotten so complicated that it really takes a village of activities, different technologies, different disciplines to be brought to bear on very complex problems. We've been doing this for many years in clinical research, and now what we are finding is that we really have to bring not just the clinical research capabilities together as we have so brilliantly here, but to also embed within that the basic scientists, the translational investigators, the technologists, and so on, that allow us to really dig deeply into the underpinnings of a process and understand how we can exploit knowledge to convert that into new diagnostics, new drugs, new policies, new educational materials that really drive differences in public health.
What diseases have you chosen for the Moonshot program?
Well, we have a number, and it's important to appreciate that this was a process that unfolded over many months involving hundreds of investigators from different disciplines. And they were charged with the idea that were it not for limited resources or challenges with respect to technology, et cetera, if they really could do what they needed to do, were they in a position to dramatically reduce mortality due to that cancer in the next 5 to 10 years? That is to say that we had enough knowledge, we had enough technology that was actionable, and we wanted to organize ourselves to execute on that knowledge and ability. And so in that regard, there were several cancers that we felt were poised for a decisive assault across this broad beachfront of activities, prevention, detection, prognostication, treatment, and survivorship. So they included melanoma, lung cancer, women's cancers, namely triple-negative breast cancer and ovarian cancer, which extract a very significant toll on women, major unmet needs, lots of great advances, tremendous opportunities. A leukemia moonshot, which includes MDS, AML, and CLL. Frankly, for CLL, we feel we're orbiting the moon already. We're very close to putting that disease in the history books, thanks to the brilliant program that Michael Keating has led worldwide. And, of course, prostate cancer, which is an enormous problem for men and continues to be. Now, it's important to stress, so those are the main moonshots, the inaugural programs. But this planning exercise, where we've analyzed all of these other cancers, has identified areas of opportunity for investment in other cancers as well. So those are also going to be receiving support, and we're going to try to elevate those programs so they too can be the moonshots of the future. And they include colorectal cancer, bladder cancer, pancreas cancer, and the most deadly form of brain cancer known as glioblastoma.
I understand that because we have a very strong leukemia program and 80% of our...
Second to none.
And 80% of our patients are in clinical trials. You said we're orbiting the moon with CLL. I understand that some of these are poised to meet some milestones almost immediately.
Well, I think that there again, we have some extraordinary targeted agents that are squarely directed at critical rate-limiting elements in the drivers of this disease. And this is something that we want to harness and exploit as rapidly as possible. The current standard of care, which has actually brought us significant cure rates, also extracts on the back end a significant problem because there are a lot of secondary cancers. With these new targeted therapies, we're in a position to not only get improved cure rates, but also to have the problems of secondary cancers be part of the history books as well.
And why MD Anderson? Why are we particularly suited for a Moonshot program?
MD Anderson has many aspects that really position it to help at least articulate and initially lead this effort. First of all, we have a singular focus on a disease. Secondly, we have a critical mass. We have teams that we can put on the field today that are extremely impressive, talent-wise, number-wise, discipline-wise, technology-wise. We have 19,000 employees, 7,000 trainees with this singular focus. We have tremendous infrastructure, clinical trials infrastructure, an outstanding clinical research enterprise, tremendous excellence in the laboratories which allow us to move in areas that are still clouded in mystery. All of these things are very important aspects that have given us confidence that MD Anderson can take advantage of existing knowledge and convert that into things that matter for patients. But I think one aspect really rises above all, and that is the collaborative spirit of MD Anderson. It is in its DNA. There are very few institutions, I think, would have been able to conduct the battle trial, which required the surgeons, the imaging experts, the oncologists, the pathologists, the genome experts, the computational scientists.
For lung cancer.
For to be able to bring that collection of capabilities and do this historic trial under the leadership of Wan Ki-hong, in my opinion, is an example, an illustrative example of how this institution sets its sight on a goal and with its capability, its talent, its will and determination, gets the job done and changes the practice of cancer medicine for everyone around the world.
And I think I was amazed, too, in my research is we have a ton of data like tissue specimens, tumor registries. I mean, it seems like we have that critical mass of data that maybe other cancer centers don't have.
Yes, I think that just the sheer volume, the fact that last year we saw 117,000 patients, performed over 10 million diagnostic procedures, over 500,000 diagnostic imaging procedures, 11,000 patients on clinical trials, north of 1,000 experimental protocols, just many INDs. All of these things say that we are uniquely positioned to move the field forward in a way that's extremely beneficial for patients. But it's also important to stress that as great as MD Anderson is, it cannot do it alone. It has a rich tradition of collaboration with other cancer centers around the country, and we fully intend to do that. And with our sister institutions in the UT system and also the Texas Medical Complex will be institutions that we hope will join us in this fight against this dreaded disease. I'm also proud, extremely proud, of our global academic program. We have 26 sister institutions that if we discover something important, we can scale to the world through this network and really impact on humanity. Because it's not about curing cancer in Harris County. This project is about dealing with a human problem around the globe.
Why is the timing right? I mean, I know we've made huge strides with targeted therapies and personalized medicine. What is the impetus for the timing of Moonshots?
Yes, well, there are a couple of things. And I wouldn't have, charged that we would do this, let's say pre-2009. But since 2009, there has been an historic confluence of technological advances across a broad front that have changed our ability to understand disease on such a fundamental level that we can lead to actionable information to think in terms of preventing and curing that disease. And so, for example, in the area of sequencing, we can now sequence for a few $100s in hours what took $3.6 billion and a decade only 10 or 12 years ago. We have the ability to manipulate genes at will using RNAi technology or cDNA libraries that are ready-made. We have the ability to genetically engineer model organisms to faithfully recapitulate different cancers and use those model systems to pressure test hypotheses, test new drugs, combination of drugs, imaging agents, biomarkers, you name it. The advances in computational science are extraordinary. the ability to essentially think in silico now exists, real decision support systems that allow us to analyze extremely complex data sets. So on the technological level, we've been handed a toolbox that now enables us to really move in ways that were not imaginable even just 3 or 4 years ago. But importantly in cancer, there's a critical transition that we've now experienced, and that is that we have clinical proof of concept. That is to say that the molecular knowledge that we've gained and how the immune system, for example, operates and how it doesn't recognize cancer, that knowledge leads to drugs such as those developed by Jim Allison, the anti-CTLA-4 molecule, that now when directed against that brake on the immune system, allows the immune system to now recognize a significant fraction of melanoma patients that have advanced disease, all of whom would have succumbed, and a significant fraction of them are now alive many years out with no evidence of disease. So we have one example like this after another where knowledge has been converted into clinical diagnostics or drugs that profoundly impact on the state of health for that individual. So we can connect the dots. It's no longer an abstraction that cancer is somehow driven by some genetics or some host-tumor interactions or some mysterious deficiencies in the immune system. We actually know a lot about what makes cancer tick, and we can now direct our efforts squarely against those aspects and develop drugs and diagnostics to truly transform the disease for that patient.
Now, you have certain milestones that you're hoping to meet. What's kind of your progress for each Moonshot? You want deliverables at like certain time points?
Again, the selection of these inaugural programs are in fact based on the fact that we feel that with existing knowledge, we can make a very significant impact on mortality within about 5 years, beginning in 5 years and extending beyond that and accelerating still further. We're very proud of the fact that over the last 10 years, we've had about 1% decline in mortality per year, even though the incidence of the disease has gone up, thanks to a lot of things like prevention, earlier detection, better treatments, and so on. The challenge for us going forward is, can we accelerate that? And that's the question that I asked. So the cancers that we've selected are ones that we feel we can have 10, 20, 30% declines in mortality beginning at 5 years and out. And then set the stage with additional discovery activities beginning now to really identify what we don't know, discern those mysteries that we've yet to uncover, and use that knowledge of the future to ultimately cure all patients of this disease. And that's the goal.
And so I guess a patient who would be in active treatment today might ask the question, what's in it for me?
Well, first of all, as I mentioned, there's a lot of proof of concept that we know that knowledge leads to interventions that have an impact on patients on the ground. And an example of this today, done on a much smaller scale, was our PREDICT trial, where patients entering into phase 1 clinical trials were evaluated for only 11 genes. And those 11 genes were genes that were actionable from the standpoint of having drugs on the shelf that we can then apply to that particular patient. About half of the patients that we analyzed in our phase 1 unit had some abnormality in one of those 11 genes. Those patients were concierged into a particular clinical trial based on those abnormalities to match the right drug with the right patient. Whereas the other patients that did not have any of those abnormalities were assigned randomly, as they normally are in phase 1, to test the dose and toxicity and so on for that particular drug. The traditional response rates in the unselected population was 5%, and that's what we saw in our study. For those that were slotted into specific clinical trials, there was a 27% response rate. So now what we hope to do in the very near future is fully sequence the patient's entire genome and use very sophisticated analytical capabilities to have a much broader spectrum of opportunities to bring the compendium of drugs to bear on that particular patient.
So what is your endpoint? Obviously to cure cancer, but what would you consider to be a success at the end of the Moonshot programs?
Well, in this initial phase, which I consider to be about a 10-year phase, I wish to, for the cancers that are selected, that we have at least a minimum 30% decline in mortality, and that we do so beginning at about 5 years. I think for certain cancers, we will do much better than that. For other cancers that are the ones not yet selected, I think we don't yet have a line of sight. But if the history of medicine has taught us and the kinds of will and spirit that we're resident here at MD Anderson prevails, that there's no question that those cancers also will fall to the axe of science and our brilliant clinical investigators.
So what about the non-Moonshot cancers, patients who have cancers other than the ones you've mentioned? What do we say to them?
Well, first of all, we continue to render care to all patients with cancers, both rare and common. But it's important to stress that knowledge that's derived from one cancer is often directly applicable to other cancers, that mutations that we may discover or drugs that we may discover in one cancer and accelerate their testing and their approval could be drugs that could be applied to other cancers very readily. We've seen this repeatedly. For example, a drug that was developed against lymphoma turns out to be very important for lung cancer and is having miraculous results for patients with that particular genetic abnormality in lung cancer. So we anticipate that the progress that we'll be making on these inaugural programs will drive progress in all cancers. In addition, the platforms that we're developing are platforms that are enabling. They're in drug development or the development of early detection biomarkers or cancer control activities, and those will be available for all cancers to help drive progress in all cancers, even the ones that did not undergo strategic planning. So if there are opportunities and chance for us to really make an impact on specific cancers that are outside of the moonshots, we will seize that opportunity. Patients are counting on us to do that.
And as far as public policy, I know you've reached out because obviously some of these cancers in the Moonshot are very preventable. So you're kind of stepping outside the research and clinical avenue to shape public policy as well.
That's correct. In the area of cancer control in particular, academically we've done really well in identifying the risk factors for cancer, publishing great papers. But then the question is, how can that knowledge be translated into folks on the ground so that they don't get exposed to those environmental carcinogens? And we do that to a certain extent, but we don't do it on a scale that truly impacts across the entire country and the world. And so let's consider melanoma. We now know that early childhood sun exposure below the age of 18, sunburn type activity, will increase the incidence of melanoma in your 30s and 40s. And so what we would like to do would be to develop the educational material for the school system in the United States, K through 12, so that we have age-appropriate, ethnic-oriented, language-enabled educational material that reaches our kids in the schools and indirectly educates the parents so that they are taken out of harm's way and that they're sun protected. Similarly, we have tanning booths throughout the United States, and half of our states do not have laws that prevent kids under the age of 18 from being exposed to excessive UV radiation. So if you wanted to solve the melanoma problem in terms of exposure, it's not good enough to publish a paper to say that early childhood sun exposure causes the disease. You've got to convert that into educational material that's effective, and you've got to have laws written that prevent kids from being exposed unnecessarily to harmful UV radiation.
Well, this is very, very exciting. As we're summing up the Moonshots program, what would be the takeaway message for both survivors, people who are about to be diagnosed, and the public at large?
We're at a very special turning point in the history of the field where for the first time we have strong technological capabilities to enable us to have science-driven cancer care from prevention all the way through to treatment that will lead to dramatic decreases in mortality in the near term and ultimately set the stage for putting this disease in the history books.
Thank you very much for being with us today. If you have any questions about anything you've heard today on Cancer Newsline, please contact Ask MD Anderson at 1-877-MDA-6789 or online at www.mdanderson.org/ask. Thank you for listening to this episode of Cancer Newsline. Tune in for the next podcast in our series.